For Chapter 10, we're going to be looking at what is referred to as cell reproduction. This is basically how one cell becomes two, whether we're talking prokaryote or eukaryote. Now, for us multicellular organisms, the cellular reproduction, where one cell divides, becomes two, is basically how we grow. You went from one cell, a zygote, to the trillions upon trillions and trillions of cells that's sitting there right now listening to me. It's how we maintain our bodies. I mean, right there, in an instant, millions of cells throughout your body just died because they hit the end of, their ability to repair themselves. So they have to be replaced. So you have to maintain your tissues. Today, I scraped a knuckle, cut open the skin. Instead of having an open wound for the rest of my life, the tissues surrounding the wound are going to fill it in with new cells. Tissue repair. So this chapter is going to be looking at how we do that. It's also going to be looking at single-cell organisms. Single-cell organisms use this cell division we're going to be talking about not for growth, not for repair, not for maintaining tissue, since they're a single cell. For them, it is truly reproduction. One cell becomes two, the next generation. Those two, given enough time, will become four, and so on. Now, it's nothing for a cell to divide, okay? There's machinery that we'll talk about in this chapter where the cell can just, pinch off and make new. The problem is, for a lot of cells, for most of the cells out there, especially the eukaryotic ones, there's more than one chromosome. So there's going to have to be steps involved to ensure that each new cell is going to have a complete set of chromosomes. The complete set of chromosomes we refer to as the genome. Our genome as humans is 32. 32 pairs of chromosomes. So, when you look at prokaryotes, though, bacteria, it's a single circular genome, singular circular chromosome of DNA. So for prokaryotes, pretty much all we're going to be talking about in this chapter has absolutely nothing to do with them. They do not have to go through all these steps to ensure that the next generation of cells has the complete set of chromosomes for the genome. Now, when it comes to prokaryotes, when they go to divide, as we'll talk about in this chapter, and if you really want to know more about it, take microbiology with me. They're also going to have smaller chromosome, smaller piece of DNA, a small circular piece of DNA we refer to as plasmids. Whereas the large circular genome for prokaryotes is going to carry, millions of nucleotides, a few thousand genes. Plasmid's going to be measured in thousands of nucleotides, a dozen or so, maybe even less, depending on the size of genes.